What is the electron transport system? A system which eventually generates ATP. How? We will see all this in detail. This is Dr. Pankaj Kumar, your mentor for today's sessions. So first of all, let's talk about that where this ETS or electron transport system is present. It is present in mitochondrial inner membrane. Okay. So it consists of respiratory molecules which are arranged according to their increasing redox potentials, right? So whatever the intermediate that has been formed in glycolysis or the Krebs cycle in the form of NADH2 or FADH2, that is being utilized in electron transport system, and eventually ATP is formed. Okay. So if we hydrolyze the inner membrane of mitochondria, altogether we can get 4 complexes. Okay. And they are complex 1, what we call NADH coenzyme Q reductase, complex 2, that is succinate coenzyme Q reductase, complex 3, that is cytochrome C reductase, and complex 4, that is cytochrome C oxidase. So all these 4 complexes participate in the entire process. First of all, we will see that how these complexes are located within the inner membrane. Have a look at the diagram. You can see there that this is the complex 1. So it is the acceptor of the NADH that is formed the mitochondrial matrix. Now the complex 2 is the enzyme membrane linked. You can see there. Then there is a complex 3 that consists of cytochrome B, C1, C, and finally the complex 4 that consists of cytochrome A and A3. And this also acts as a terminal oxidation. Okay. Now here you can see 2 things. One, the 2 electron acceptors. One is a coenzyme Q and second is a cytochrome C. Okay. These are mobile carrier. So they are not fixed. Rest of the complex 1, 2, 3, 4 are fixed. They are not movable. But as far coenzyme Q and cytochrome C is concerned, that is a mobile carrier. So just keep the very position of coenzyme Q in your mind, and that is, it is present between complex 1 and 3, that is the coenzyme Q, and the cytochrome C is present between complex 3 and 4. And they being, since there is a mobile, so what happens? They carry on the electrons from the cytochrome C1 to finally to cytochrome A3. Okay, one more thing you need to keep in mind, that this is present at the upper layer of the membrane, not at the lower layer, right? Cytochrome B is always present at the lower layer. So the relative position of the molecules is equally important. Let's see how the process occurs. Now electron transport system, we see altogether there are 6 electron acceptors and they are arranged in the given sequence like FMN, coenzyme Q, cytochrome B, C, A, A3. You can see there FMN, coenzyme Q, or that is also known as ubiquinol, right? Then cytochrome B, then cytochrome C, and cytochrome A and A3. Okay. So what happens? Electrons are arranged according to their increasing electron affinity. So we can say that the very first complex has the least amount of electron affinity, and the last complex, that is the complex 4th, has the highest amount of electron affinity. Okay. So this arrangement actually enables the flow, continuous flow of electrons. So how it happens? Let's try to understand. You see, this is a matrix and here NADH and H is formed. So what happens? The very first complex, that is NADH, they accept it, and what they do, they transport the H ion to the perimitochondrial space, and electrons are being transported inside the membrane. Okay. So this electron is accepted by FES, finally the ubiquinol, right? And eventually it goes to the terminal oxidations over here. Okay. So electrons are being carried in the membrane, but in the process what happens, that H ion being expelled in the perimitochondrial space. So for NADH it is the site. For when the electron passes through ubiquinol, that another 2H can enter over there. Similarly, if they passes through the cytochrome C, then again another 2 hydrogen can enter over there. Okay, so these are the pathway where what happens, that hydrogen ion being pumped in the perimitochondrial space. So why they important? Because what happens, the objective is to generate an electrochemical potential difference between the perimitochondrial space and the matrix, so that it will help in the generation of ATP. We will see all these things in Michael chemoosmotic hypothesis, right? So what happens that during the ETS, the NADH2 gives 2 electron or 2 hydrogen to FMN, and thereby 3 ATP are generated. So one NADH2 is capable of forming the 3 ATP. But when we see the FADH2, the FADH2 give only 2 electrons. Okay. So what happens here? Only 2 ATP are formed during the entire process of oxidative phosphorylations. Okay. So please remember, NADH is capable of forming 3 ATP and FADH2 capable of forming only 2 ATP. Okay. Now let's talk about some of the inhibitors of electron transport system. Many a time this question has been asked. So please remember, rotenone and amytal, that is an inhibitor for complex 1. Antimycin is a inhibitor for complex 3, and cyanide or carbon monoxide is a inhibitor for complex 4. Okay, so that is very, very important. So please remember this, right? Let's talk about the Michael chemoosmotic hypothesis. Okay, so right now we have seen that during the electron transport system, the electron being transported through the membrane and the hydrogen ion being pumped in the perimitochondrial space. As a result, what will happen? That perimitochondrial space will become more acidic, or the hydrogen gradient will be produced. Okay, the entire membrane is impermeable for the passage of the hydrogen. There is only one place where it can pass, and that place is what we call ATP synthesis. Okay. So this is a knob-like projection in the matrix. It consists of 2 units, one what we call F1 particle that contains the catalytic site for ATP synthesis, and there is a F0 which serves as a transmembrane for the channel for hydrogen flow, right? So the F1 particle enables the coupling of ADP and inorganic phosphate forming ATP. Okay, and this F1 and F0 complex serve as the molecular apparatus for coupling of H movement to the ATP synthesis, right? So have a look at this. So what we are seeing, that due to electron transport system, this H ion is actually being transported in the perimitochondrial space, and due to there is a higher concentration of H ion with reference to the matrix, so it will the electrochemical gradient wants that the H ion should come inside, and as a result there is only one place where it can come, that is ATP synthesis. When it comes, what happens? ATP energy is released, and that energy is utilized for the coupling of ADP and phosphate forming the ATP. So this is how the oxidative phosphorylation happens, and that is going to work in the similar manner in the case of respiration as well as the photosynthesis. Have a look at this diagram. So you can see there that this is a F0. So this is the F0 particle that is providing a membrane for, or we can say pore for the transmission of H ion, right? And this is the F1 particle, the terminal portion that catalyzes the binding of inorganic phosphate and ADP forming ATP. Okay, so this is catalyzed by another enzyme, and the name of the enzyme what we call ATP synthetase, right? So this is how the entire things works. So the ATP is generated inside the matrix, but the requirement of ATP is outside the mitochondria for the living beings. So how it is done? It is transported from the matrix of the mitochondria to the cytoplasm by ATP ADP translocase that is present in the membrane. Okay. So what happens? ATP and ADP cannot freely diffuse in the mitochondria. Both are linked. So the exit of ATP is coupled with the entry of ADP, because ADP is required so that further ATP can be formed. So that's why with the help of translocase, what happens when ATP goes out, ADP comes in. So this is how the entire the balance between ADP and ATP is maintained within the mitochondria, right? Now let's talk about that how it is being regulated. Okay, electrons do not flow unless ADP is present for phosphorylation. So this is one of the regulatory methods. Okay, and increase ADP level causes an increase in the activity of various enzymes like glycogen phosphorylase, phosphofructokinase, citrate synthetase, and we have already seen that higher concentration of ATP is going to inhibit them by the allosteric inhibition. We have seen in case of phosphofructokinase also, we have seen in case of citrate synthetase also. So it is the concentration of ATP which eventually regulates the oxidative phosphorylation as well, right? Let's talk about the energetics. So in case of glycolysis, what we have seen, the formation of 2 ATP, right, and 2 NADH2. This 2 NADH2 is coming to oxidative phosphorylation in case of mitochondria. Okay. Now this acetyl coenzyme A also forms, during the formation of acetyl coenzyme A also form 2 NADH, and Krebs cycle is going to form 6 NADH and 2 FADH2. So altogether we see that 36 to 38 ATP is formed. Okay, so it's 36 in one case, 38 in another case. We will see about that, right? So have a look at this. In case of glycolysis, what we have seen, 2 ATP, 2 NADH capable of forming 6 ATP. Mind it, this 6 ATP can be formed only when NADH will enter into the electron transport system. So there are two pathways by which it can enter. Okay, one what we call glycerophosphate pathway. In that what happens, there is a loss of 2 ATP, so 4 ATP is formed, and that is happening in muscle and brain. But in case of heart, what happens, that there is a different shuttle, what we call mallet shuttle, and in that mallet shuttle, instead of 4, 6 ATP is formed, then total amount of is a 38 ATP. So the difference occurs only due to the accommodation of glycolytic NADH2. Okay, so let's be very clear. In case of heart, 38 ATP. In case of brain and muscles, there is going to be 36 ATP. Now let's discuss how this glycolytic NADH enter into mitochondria. I told you there are two pathways. Okay, one is what we call glycero 3-phosphate shuttle, and second what we call malate aspartate shuttle. Okay, one by one we will see. So let's first of all see glycerophosphate shuttle. Now have a look at this shuttle. You see here what happens, that NADH is going to bind with a DHAP, dihydroxyacetone phosphate, and as a result what happens, glycerol 3-phosphate is formed and NAD is formed. Okay, now this glycerol 3-phosphate enters, is again bind with a FAD and form FADH2, and FADH2 can directly enter into the Krebs cycle, you know very well, but FADH2 will produce only 2 ATP instead of 3. So so one NADH is going to produce 2 ATP by this particular pathway, and we have already seen this pathway is going to occur in case of brain and muscle, right? But in case of malate shuttle, what happens, that the NADH is actually combined with oxaloacetate and malate is formed, and malate directly enter into the mitochondria, and you know very well, when there is a one amount of malate, then malate will form two, when malate will get converted into oxaloacetate, then one molecule of NADH2 is going to be formed, and one NADH2 is capable of forming 3 ATP. So in that case what we see, that altogether 38 ATPs are formed, right? So this is all as far as the malate aspartate shuttle and the glycerophosphate shuttle is concerned. I hope the reason for the difference in 36 and 38 ATP is very clear. That's all, as far as today's session is concerned. Thank you.